Hello and welcome to another episode of Momento Malum today. Plant breeding has made a profound impact on food production by improving the economic, quality, and productive capabilities of the specific crop in question. The durian is no exception to this rule. The durian is a crop of Southeast Asian origin. It is commonly known as the king of fruits within this region. In the past, attempts were made to determine the genetic diversity of the durian gene pool via morphological markers, but is found to be ineffective. Since then, the work has shifted to genetic markers, and multiple papers have been dedicated to determining the genetic variation within the durian gene pool. For this video, we'll be covering 4 papers dedicated to the topic of durian genetic diversity. The first video we'll cover is by Dr. Mursyidin, who analyzed the genetic variation of 11 durian cultivars of Borneo. This was done using rapid marker technology. The data suggests that these 11 could be broken down into 6 groups, which can be seen on the next slide. Group 2 was found to be the most genetically similar, while groups 5 and 6 were the least genetically similar, and had the greatest level of genetic diversity. Another paper, this time based in China, was written by Dr. Lin, who utilized SSR marker technology to analyze the genetics of 32 Chinese-grown cultivars. Using this technology, the gene pool was broken down into 2 distinct genetic groups. The first consisted of 10 genetic lines. The second group was made up of the remaining 22 cultivars. These 2 will be listed on the next 2 slides, but the lineage of the current cultivars seems to be derived from 2 populations. 15 of the 22 lines of Group 2 show minor genetic traces of Group 1 pedigree. The 10 lines from Group 1 and the 7 lines that remain in Group 2 seem to be genetically pure in nature. The third paper was based in Indonesia and was written by Dr. Hariyati. In the mid-2000s, another durian species was analyzed. This was known as the pakin. This species had a flavor of the durian without the trademark rotten smell of the regular durian, although the amount of fruit in the fruit was far less than a common durian. As a consequence, there's various interest in crossbreeding both species to get new varieties contain the best of both worlds, and this process occurred in various breeding programs. 19 durian lines were analyzed using rapid markers. These include 1 pakin line, 2 pure-blood durians, and 16 hybrids. Of the 16 hybrids, these cultivars were grouped into 2 distinct groups. Group 1 consists of the cultivars UB2, UB16, and UB22. The rest fell into Group 2. The family tree will be seen on the next slide. The data found that the crossbreeding between the pakin and the durian produced high levels of genetic diversity in the hybrid lines. But it was also found that the durian proper contributed very little to this process, and most of the variation is derived from the pakin lineage. The lineages of Group 1 were found to be the most genetically divergent of these populations, and can be used in the breeding of new cultivars. The last paper we will cover is another Indonesian paper written by Dr. Mursyidin once again. This researcher would analyze 18 more cultivars from Indonesia using the RBCL genetic marker, and here are the results. The genetic data found that the 18 cultivars could be grouped into 4 groups. This will be shown on the last slide. Many of these cultivars belong to different species, and as a result, the specimens are quite divergent in genetics. The researcher hopes that crossbreeding between these species could aid in the breeding of new cultivars. The data will be shown on the last slide. Now onto the last slide, which shows off the 4 groups that the 18 cultivars could be grouped into. And that about covers everything. Thank you for watching. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below.